Hello and welcome to my channel. So in today's video we are going to discuss about protein techniques, the techniques which are involved in protein isolation. So basically protein purification is a series of processes intended to isolate and purify a single protein or complex from cells, tissues or whole organisms. And separation steps usually exploit differences in protein size, physical chemical properties, binding affinities, and biological activity. So, protein purification is either preparative or analytical. Now, there are two types of methods which we can use to isolate protein: preparative quantification method and analytical method. So, in preparative quantification, it aims to produce a large quantity of purified proteins. It's a preparatory method. Whereas in analytical method, it produces a relatively small amount of protein for a variety of research, including identification of protein and study of protein, its structure and function. The first step is sample processing. So, if the protein of interest is not secreted by the organisms into the surrounding solution, the first step is disruption of the cells containing the protein. So, we'll just break open the protein, the cell, so that we can get the total amount of protein. The process is called homogenization. Second step is centrifugation. So it is a process that uses centrifugal force along with gravitational pull to separate mixtures of particles of varying masses. So we can see here this is a centrifuge tube in which the protein samples, they are being centrifuged. The protein is suspended in some solution, and then after centrifugation we can see the supernatant and pellet, they have been separated from each other. There is a supernatant which is liquid and pellet is a hard semi jelly like material. So when a vessel containing a mixture of proteins is rotated at high speeds, the inertia of each particle yields a force in the direction of the particle's velocity, that is proportional to its mass. For precipitation and differential solubilization, so this is used in bulk protein purification. It's a common first step to isolate proteins is precipitation using a salt such as ammonium sulfate, and we call it salting out. So proteins are precipitated by ammonium sulfate, NH4 whole twice SO4. And in the next method, we do dialysis. The process of dialysis separates dissolved molecules by their size. So the biological sample is placed inside a closed membrane where the protein of interest is too large to pass through the pores of the membrane. So we have a dialysis bag over here with pore size which is very small, through which the large macromolecules cannot pass through, but the small contaminants, and if there are some extra ions, they can only pass through. So there is a cutoff of these dialysis bags, the pore size, which retains the large proteins molecules and macromolecules inside it. The next method is chromatography. So it is used in almost all protein purification methods and allows the separation of a given protein from thousands of different proteins in cells and tissues. So there are four main types of chromatography. First we have gel chromatography or molecular sieve chromatography. We also call it size exclusion chromatography. So chromatography in which proteins can be separated according to their shape, size or molecular weight. It's called size exclusion chromatography. So here we can see that we have a column with jelly material inside, and this gel material has pores through which our protein sample can pass through. Now let's assume we have a sample with different masses and molecular sizes and shapes, and now what will happen, the pore size and the size of the protein, if the size of the protein is big, it won't be able to enter inside the beads, the pores, and it will just pass through the void volume and will come out of the chromatography column immediately, because it's not even the part of the competition. And in fact, the very small ones will not be able to enter inside the beads. They will just go in and come out. So now the real competition will be between the intermediate sized protein samples which are able to enter inside the beads, the pores of the chromatography column. And then when they compete, the one which are small in molecular size, they'll be able to come out first, elute out first, and the rest will follow. So in this way, the highest molecular weight sample will come last, and the one which is small in size will come first. So on that's how separation of molecules on the basis of size will take place. Next, we have ion exchange chromatography, in which separation takes place on the basis of charge. So the column to be selected is according to its type and strength of charge. So we have anion exchange resins, which are called negatively charged compounds, and in cation exchange resin, they have a negative charge and are used to separate positively charged molecules, the cations. So this is an example of cation exchange column. So we have a column over here with beads containing negative charge. Now the negative charge beads are obviously able to attract the positively charged protein sample. So if we have a sample with protein sample with positive, negative and neutral proteins within it, and we want to differentiate, so the beads containing negatively charged functional groups, they will be able to bind positively charged proteins, and later on we can recover these positively charged proteins from the column beads by either changing the ionic strength of the column or changing the strength of the buffer. So before loading the column with protein, the negatively charged beads would interact with positively charged counter cations, and when the protein solution is introduced to the column, the positively charged protein will exchange with the bound sodium ions. Now an ion exchange consists of positively charged beads which exchange bound anions. And proteins bound through ion-ion interaction, they can be eluted by increasing the sodium concentration in the eluting solution. Next, we have affinity chromatography. It is a chromatography in which proteins are separated based on binding to a ligand covalently attached to a column bead. And the chromatography resin along with a group binds to a specific site on a given protein of interest. So it may be a group that binds to the active site of an enzyme, and it is a powerful technique since antibodies can be formed that will bind selectively to a single protein. So here we exploit the property of the biological property that certain molecules have affinity towards each other. Next, we have HPLC, high performance liquid chromatography, and fast protein liquid chromatography. So it is a form of chromatography applying high pressure to drive the solutes through the column faster than using gravity forced flow through the column. Now, protein purification is necessary to have a quantitative system to determine the total amount and concentration of the target protein and total protein at each step. So now let's understand what is total protein first. So it is calculated by measuring the concentration in a fraction of the sample and then multiplying that by the total volume of our sample. Let's say we have 7.5 microgram per microliter of protein with us. That means that we have 7.5 microgram in 1 microliter of solution. That is 75 microgram in 10 microliter, which is equivalent to 75 milligrams in 10 ml. So we can call that we have 75 milligrams mg of protein in 10 ml. Next, we have total activity. It is measured as the enzyme activity within the assay multiplied by the total volume of the sample. Example, initial sample volume is 5 microliter, let's say, and if we calculate the activity in our assay, 2.5 units per microliter it would be. So this would be equivalent to 2500 units per ml. Next is enzyme unit. It describes the enzyme's catalytic activity. So 1 unit, that is mu mole per minute, is defined as the amount of enzyme that catalyzes the conversion of 1 mole of mu mole of substrate per minute under the specified conditions. Next, we have yield, which is a measure of the biological activity retained in the sample after each purification step. Specific activity is total activity upon total protein. In our example, it was 2500 over 75, which comes to 33.3 units per milligram. Next, we have purification level. So it evaluates the purity of the protein of interest by dividing the specific activity, calculated after each step by the specific activity of the first purification step. So that's all, and rest we'll be discussing in next part of our video. Stay tuned and thanks for watching.